as you go up.
Yeah, that makes sense now.
So collagen looks like this under electron microscope. At a molecular level, it looks like that. Skip something? No. Okay, so there's several types of collagen found in different places. Like tendons and skin have the so-called type 1. Of the 3 chains of that type 1 collagen, 2 of them are the same. That means they come from the same gene. So to make type 1 collagen, you need 2 genes to make 2 different kinds of polypeptide chains, and you'll use 2 copies of one and 1 copy of the other. The kind of collagen that's found in cartilage comes from 1 gene. You use 3 copies of the same polypeptide chain. Mutations in this gene lead to osteoarthritis. And then type 3 that's found in the blood vessels to keep them strong also has 3 identical chains. So a single gene there, a different gene from that one. Collagen is really weird. It's the protein that's found in Jell-O. It's not a very good protein nutritionally for this reason. It doesn't have very many different kinds of amino acids. It's about 1/3 by composition of glycine. 11% alanine, and proline and all of its cousins add up to about another 1/3. So between those 3, you're like 75–80% of the whole protein tied up in these 3 types. It's got derivatives of proline that are hydroxylated. This is 1, 3-hydroxyproline, 4-hydroxyproline. And it's got hydroxylysines. You don't see these in the standard 20 amino acids that we talked about. These are created after the fact. You build the polypeptide chain up into a protein, and it's built with prolines, and then another enzyme comes along, sees that collagen molecule, docks to it, and hydroxylate. This is so-called post-translational modification. You're modifying the chain after you've done the translation on the ribosome. So there's no genetic code that codes for hydroxyproline. Only proline is built into proteins. The modifications come after the protein has been assembled. The enzymes that do that are, there's a lysine hydroxylase that makes that guy, and a prolyl hydroxylase that puts those modifications on proline. So between the underivatized regular proline and the hydroxyproline, you add those together, another 30% of this protein is made of those. So very few amino acids go into the making of collagen. So nutritionally, you eat Jell-O, you get protein, but you only get just a handful of amino acids. It has this special sequence, glycine XY, repeated over and over again. If you march down the chain, every 3rd residue without fail is a glycine. And it's because of structural requirements. It's a keeped up little helix, and you wrap 3 of them together. If you had a bulky side chain sticking out there, it wouldn't wrap around each other very good. So glycine is used when they come together in the center positions here. If you take that molecule and turn it on its edge and look down the pipe, the glycines are all in the center. They have the hydrogen R groups. It's the smallest R group, so you don't get any steric crowding from that. That's why you have this glycine XY. So these X's and Y's would be the alanines and the prolines and things like that.
What's the reason it can carry it so easily? Like, and
What happens
like,
usually?
what